A simple and straightforward means for discrimination is to evaluate the intensities of the peaks from the spectra, particularly to look at peaks that have some meaning for the minerals involved. For example, in amphiboles, sodium and calcium, magnesium and iron compete for similar sites within the structure. So looking at ratios of their intensities might give us clues to differences between the populations. These are seen here. The individual granites from which the amphiboles came from are shown in the red figures across the diagram. For comparison, I've also plotted the peak intensities for some known and well-characterized amphiboles in the various colors on the legend at top. What we can see here is there are specific ranges in which the populations fall, and that might be useful for telling us the differences between these amphiboles. A more complicated technique for resolving differences is principal component analysis. Here we use the entire spectrum from one grain and compare it to a spectrum from another, and another, and another. Those with similar spectrum should plot close together on this graph. Those with vastly different characteristics should plot further apart. The gray dots in the background even tell us which peaks are participating in moving the differences, and their directionality and intensity tells us how different those peaks are. Principal component analysis can allow us to say that certain of minerals are very similar to one another, while others are vastly different. Again, we struggle with the problem of knowing exactly what those differences are, but we can start looking at things that are comparable to one another with the LIBS. So the LIBS uses UV light in the form of a laser and induces visible and near visible radiation, which is characteristic of the atoms involved. And it's a tad messy. To get away from the messiness, we can work in the higher energy part of the spectrum, X-rays. The trade-off is that we need more specialized conditions to pull this off. I want to start with the general process of X-ray fluorescence and actually introduce a machine that is designed to look at the fluorescence generated by a sample. So this machine is diagrammed on the right in a cartoonish fashion. You need an X-ray tube. The X-ray tube emits X-rays of a certain radiation, a characteristic radiation. These then hit a sample. The sample then fluoresces its own radiation based on the elements that are in it. These are then sent to a crystal with a known D spacing. Recall that in XRD, we sent X-rays through crystals that we didn't know the D spacing to find that out. But here we have a custom-made crystal that we know the D spacing very well. So we can use the Bragg equation, but what's missing from the Bragg equation this time around are the wavelengths of radiation. We don't know the source. That's in our sample. It was induced by the known characteristic X-rays that we sent to it. We can then collect the X-rays at various angles and know what particular wavelengths respond to what particular elements, and assess them that way. The X-rays generated by this technique tend to land on a 3-diameter circle, so it works best for large amounts of material. These materials have to be powdered and either put into the machine as a powder or fused into a glass for the analysis to work. However, the analyses are incredibly accurate, quantifiable, down to the parts per million level. Generally, we use this for analyzing entire rocks and not individual mineral specimens. However, with a large enough specimen that you're wishing to destroy in the process, you can make a very accurate analysis of its composition. What we are characterizing in this technique is the fluoresced X-rays from the sample at the wavelengths appropriate for the atoms that are in the sample. So it requires looking at the spectra of energy that are released from the sample in the form of X-rays. The technique that works best for quantification is the resolving of specific peaks. This is known as wavelength dispersive spectrometry. That is, characterizing the peak height over background so that we understand the nature of the intensity that's being released by a specific characteristic radiation. Usually the strongest one, but sometimes other ones are necessary. The only way this technique can work is if we know what the peak should look like from a material that is similar, that has the atoms of interest in it. So we need a standard. Wavelength dispersive spectrometry works because we can compare a standard that has been analyzed by a number of different ways, a number of times, and then compare our unknown sample against it and make quantification determinations based on the peak differences and some differences in the material itself. If we want to analyze a smaller volume of material, say a small mineral grain or even part of a mineral grain, we're going to need a much more focused technique. Turns out that X-ray fluorescence happens in the electron microscope. Electron microscope simply puts a focused beam of electrons on the surface of a sample. In doing so, it causes the same fluorescence that we've seen by applying X-rays to the surface. So by generating X-rays in a very small volume of our sample, we're able to send them through a crystal of a known d spacing, which can then be targeted at an X-ray detector, and we can look again for the characteristic radiation for individual elements of interest. This is the more common technique used for characterizing mineral compositions in a quantitative fashion. Electron probe microanalysis, or EPMA, simply is applying wavelength dispersive spectrometry with a source that's much more focused. The nice part is we can do micron sampling, actually down to a single micron. You can get resolution in terms of hundreds of parts per million on most instruments, and it's relatively non-destructive. There may be a little bit of scoring from the electron beam, but that's about it. The preparation for the instrument is somewhat difficult, but not terribly onerous. Samples have to be flat and well polished, and the entire system operates under a vacuum, and that may take a while to achieve with some samples. Again, we're just applying the Bragg equation. This time, again, we don't know the wavelengths we're generating. We're after the elements of interest that are producing those wavelengths. Much like X-ray fluorescence, EPMA relies on wavelength dispersive spectrometry to produce accurate analyses. That means we're after the peaks in their locations for characteristic radiation from an element within our sample. We're characterizing the background that surrounds that, and we're comparing it to a known standard on a frequent basis. The known standards have to be characterized through other means or by multiple analyses so that they are useful for speaking to the elemental composition of our unknown material. It is possible with the right detector to see all of the energies that are being emitted over an entire spectrum for X-rays. This is known as energy dispersive spectrometry. The trade-off is that it's difficult to make a highly accurate quantitative analysis using this technique. However, you can determine which elements are present underneath the electron beam and make some judgments of their relative concentration. For example, if you look at this spectra on the left, you'll see that there are strong peaks for iron and titanium and oxygen. There are also peaks for carbon, aluminum, and silicon. One thing to note, though, is we coat the sample in carbon so it can conduct electricity. So this carbon has nothing to do with our mineral. It is simply a thin film on top of it. However, the other elements are real and producing X-rays. The high levels of iron, with a lesser amount of titanium and a relative amount of oxygen that's proportional to the iron, tells me that we're looking at a magnetite grain that is rich in titanium. Not unusual for the rock we were looking at. This is a fast way to determine what elements are present, and usually robust analyses take 2 to 3 minutes, whereas it could take a lot longer to standardize for wavelength dispersive spectrometry. Because EPMA is based on electron microscopy, we can get images from the instrument. On the left, we have an example of secondary electron microscopy. Secondary electrons are those electrons that are kicked out of atoms when they are hit by the electrons from the gun. At the energies of most electron guns, this only happens in the first 50 nanometers of the sample, and therefore it gives you surface topography. It's not very interesting for a flat sample, but for a sample with some relief, it can show some dramatic things at a scale beneath that of an optical microscope. For example, the picture we're seeing here is a volcanic foam generated in Hawaii from an eruption. Volcanic bubbles pushed small amounts of magma to the outside edges, leaving them as an interconnected glass upon cooling. And that's what we see here, a three-dimensional framework of the glass left behind around the bubbles. On the right, we have an example of backscattered electron microscopy. Backscattered electrons are those electrons from the gun that hit the electron clouds of the atoms in the sample and bounce back out. They are fairly high energy, and the intensity of these is wholly dependent on how the electrons are structured in the sample. Most basically, the heavier elements, the higher Z elements, reflect back more electrons than the lighter ones. So there is compositional information in these figures. They are grayscale, as electron images are, but the brighter grayscale on this image belongs to grains of magnetite, whereas the lesser gray, particularly the grain off to the right, belongs to a feldspar. The black in the background actually belongs to a glass that has a much lower mean Z. So to conclude, we've used the fluorescence of light from atoms to determine which atoms are present in our minerals. The frequencies are emitted at characteristic wavelengths and energies, so we can figure out exactly what we're looking for by looking at energy or wavelength spectra. The key is to get the electrons moving. Excite the atoms, get them hot, and then look at the resulting light. The techniques we looked at were the laser-induced breakdown spectrometer, which is quick but produces hard to quantify materials and is somewhat destructive in that it leaves a hole in your sample. We looked at X-ray fluorescence, where X-rays excite a large amount of material to produce highly accurate data, and we looked at electron probe microanalysis, where electrons are targeted on a sample to produce X-rays that can then be characterized. XRF and EPMA rely on wavelength dispersive spectrometry to make accurate analyses, and they also require standardization for that accuracy. What we get out of this is compositional analyses of our materials. What elements are present, and in the case of quantification, how much of each are there. This is very different from X-ray diffractometry, where we are interested simply in the structure of the materials. In other words, how the lattice is put together. If you need to know which elements are present in your sample, you need to turn to one of these techniques to understand what your sample is made out of. It does nothing for polymorphs, right? Because polymorphs have the same composition, but it will tell you what elements are present and which polymorphic set you should be looking at. This concludes the lecture on X-rays and lasers as a modern technique of evaluating minerals. Thank you.